Okay, we're moving on to our second part. I got our kitty Lily here. You'll find out that I'm a big pet fan. So this is one of our little pets that we have at the Albano household, and she's crazy, as you can see. Okay, let's stop that one. Okay, let's start with video 2. Our little friend Lily is here to visit. As you can see, she's a little crazy. Let's get going now on genetics. We're going to be talking about cats later on this semester, female cats specifically. They're really fun. Cats are fun. But anyways, let's move on. We'll get to that in chapter, I think, 4. All right, so we started off on our first 10 slides talking about basic macromolecules. Now we're going to be moving on to what DNA is. Now, DNA itself uses the 4 nucleotides A, T, G, and C to generate a code. So, for example, ATG, GGC, this would be a DNA sequence. This is the primary sequence of a gene. When this sequence is broken up and divided into units of 3, we're going to be calling these units of 3 codons. So like ATG is an individual codon. We'll get to that again later on in the semester. When the DNA sequence is read and transcribed into proteins, proteins can also be called polypeptides. One of the things that was very confusing to me as an undergraduate is what's a protein, what's a polypeptide, what's a peptide? Here's the upshot of the whole thing. A protein is a large polypeptide. A peptide is a little tiny protein. So the way you could think about it is that a peptide is anything under 99 amino acids. Anything over 100 amino acids would be a polypeptide or a protein. We'll get to that again a little bit later. I keep promising you all this stuff that we're going to be doing later, but just trust me, we'll get there. So this DNA sequence codes for ATG, which is methionine, GGC, which is glycine, and so forth. So gene expression is basically converting DNA information into protein information that the cells understand and which allows the cells to do what they need to do. And that happens in 2 steps. Transcription, where DNA is converted to RNA or ribonucleic acid. So ribonucleic acid is like DNA's cousin. And then RNA is translated into proteins. And this process happens in different parts of cells. In a bacterial cell, it all happens in the cytoplasm. So DNA will be transcribed into RNA, or we also call that messenger RNA. And then that messenger RNA is translated into proteins. Now, I know this looks like a big glob of butter, but assume that this large glob here is a long string of amino acids that coils around to make a big shape. We'll get to that again later. So the function of the protein has everything to do with its shape, and its shape is determined by the original gene sequence. So what a cell does depends on the proteins that it makes and what those proteins do. So as long as you have proteins being made by genes, and those genes are being expressed, the cell will do whatever those proteins are telling them to do, or whatever those proteins enable the cell to do. And so the traits that an organism has are literally controlled by the individual genes that an organism has. So living cells have lots and lots of genes. In fact, the human genome has about 30,000 genes. They are not all being transcribed and turned into protein at the same time. In fact, some of them have been silenced after you were born. Some of them won't be turned on until you get older. So the genes that you came into the world with were given to you by your mom and dad, and when they're turned on, they're able to make proteins that do specific jobs. Now, all of our DNA is packaged into what we call chromosomes. Chromosomes are basically a complex of DNA and proteins. All right, what does the word complex mean? Complex is a chemical term that means you're sticking things together. You're sticking molecules together. They stick together to do so for a reason, okay? And so in the case of a chromosome, you have DNA and what we call packaging proteins or histone proteins, structural proteins. And so the average human genome or chromosome contains millions of nucleotides, that get wrapped around into these little coils and eventually are packaged in to 46 chromosomes in humans. Other animals have different numbers of chromosomes. Dogs have 76. Fugu, which is a pufferfish, I think they have like 42. The number of chromosomes that an animal has doesn't mean that they're more complex. And the amount of DNA that an animal has or that an organism has doesn't mean that it's the most complex organism. So one of the things that you need to kind of get used to right now is that chromosome number and DNA content may not predict complexity. In the meantime, let's go back to how chromosomes are packaged. What we're looking at here in this picture is an average plant cell. Here's the cell wall. The little green globs in there are chloroplasts. Chloroplasts are really interesting because they actually carry their own DNA. The little red things over here, like that one and that one, those are mitochondria. They also have their own DNA. And right here, this big purple thing is the nucleus. Nuclei, or the nucleus, are found in what we call eukaryotic cells. Eukaryotic cells package and sequester DNA inside of a central space, kind of for protection and organization. When you look at individual chromosomes, chromosomes are really tightly wound clusters of DNA with their packaging proteins. When you uncoil and unwind a chromosome, DNA is a double helical molecule with the actual code inside of the long ribbons, which are the sugar phosphate backbones. So basically what you're looking at here are the bases, C and G, this is what they actually look like, associated with the blue ribbons, which are deoxyribose sugars, and phosphates. And so basically, in human cells, what the cell has been able to do is to overcome this massive packaging problem. Inside of an individual cell, so this would be an animal cell, let's say a human cell, a cheek cell. Inside of the human cell, the nuclear space contains all 46 chromosomes, or we can also say 23 pairs. Our cells have found a way to package 46 individual chromosomes into a really tight space that needs to be highly regulated. And this is a space that you can't even see with the naked eye. And so this packaging problem has led to the ability for cells to not only regulate individual chromosomes for gene expression, but to coordinate a chromosomal dance, if you will, when a cell is dividing, so that each new cell gets an exact copy of the original set of chromosomes that the original cell had. Again, that's coming. So since DNA is such an important molecule, it's really important to understand how DNA works, how cells work in organisms, and how DNA and cell function are linked. And that was kind of the basis for the Human Genome Project, where a number of labs determined the entire sequence and order of nucleotides, or A's, T's, G's, and C's, in each chromosome. So the goal was to find out how many nucleotides were within the genome, how many genes were within the nucleotide sequence, and how are the chromosomes packaged and expressed. And some of this work is still being worked out today. Scientists from all over the world participated in this venture, and understanding the sequence can help us understand certain human diseases. So for example, here is a picture or a graphic of chromosome number 4. On chromosome number 4, you're seeing a number of diseases that are associated with chromosome 4 when certain parts of chromosome 4 are mutated. So Huntington's disease, the sequence, DNA sequence for Huntington's disease, when it's mutated, you can find up here near the top part of chromosome 4. C3b, inactivity deficiency. A number of different types of deficiencies are found down here on the lower portion of the chromosome. Normal, or I guess what you would say a wild type gene sequence, gives you a normal trait. If you do not have a wild type sequence, or if you have, let's say, a gene mutation, you may end up with one of these genetic diseases. And so to understand genetics, and in particular DNA science, we've been able to figure out ways to manipulate DNA so that we can understand how each different organism has a different set of sequences that leads to making that organism different. We're also able to look at DNA, what we call DNA fingerprints. This is a really common tool now used in forensics. This allows us to identify an individual DNA sequence from, let's say, a blood sample with something else, let's say something that was found at a crime scene. Very easy nowadays to match DNA from a crime scene to a set of suspects to say, oh, these people are innocent, please let them go. Another thing that's been very interesting in the last about 20 years is mammalian cloning. This is a really kind of controversial subject. This is where you take the DNA of one organism and you stick it into the developing egg of a young organism of the same species, and that allows you to basically make a genetically identical individual of the donor organism. The cool thing about this is if you could make this work, you could, in theory, let's say, culture cells to generate cells to help somebody who's, let's say, had a massive burn. You could grow new cells and the person wouldn't reject that tissue. The fear is that humans would be able to clone other humans. And so this has led to a lot of political debates and philosophical debates regarding whether or not DNA science should be able to clone organisms, especially humans. This is a picture showing Dolly the sheep. Dolly the sheep was the first cloned animal, successfully cloned animal, and Dolly the sheep was born of this black-faced ewe, which is a female sheep. Dolly the sheep, the donation cells came from a white-faced ewe. And so this particular organism was genetically identical to the adult that donated the tissue, the DNA originally. Sadly, Dolly died early. They had to put her down because she had arthritis and she was suffering. And cloning is just a very difficult thing to do. Many clones are born weak. They're born kind of sickly, and so it's a difficult process. I'm going to stop again now, and we're going to move on to the last video after this. So hold on tight. We'll finish this up in just a few minutes.